Hello and welcome to another episode of Momento Malum today. Near the conclusion of the First World War, the group known as the Mezzers Crossland Bros laid the foundation of sweet pea breeding in the region of Cowichan Valley. These people had been members of the National Sweet Pea Society while continuing to manufacture woolen products within Yorkshire. The production of sweet pea seeds increased in 1919 and further increased in the year after due to an increase in contracts. The trend went down in 1922, leading to Dr. Fleming attempting to document the history and work out the genetics of the sweet pea in 1925 under the University of British Columbia, Canada. It is through this paper that this video was derived. Let's get started. The sweet pea species is native to Italy. In 1699, a monk by the name of Mr. Cupani sends some wild sweet pea seeds from Sicily to Middlesex, England, via Dr. Uvedale of that region. He also sends seeds to Mr. Coleman of Amsterdam. These seeds would become the foundation of the sweet pea cultivar populations. These were at first only grown as a curiosity by interested individuals until an English nurseryman by the name of Robert Fuber in 1730 released these seeds to the general public as part of his business. The first cultivar released was known as Cupani's Original and was a bicolor cultivar with a reddish-blue outside and a purple center. The first three cultivars released were the above-mentioned Cupani's Original, the cultivar Painted Lady, which had a red center and a white outside, and the pure white cultivar aptly called White. All three were common products by 1731. The early improvements were first catalogued at the nursery of John Mason, which was published in 1793. Amongst these lines, he offered the first pure red, purple, and dark purple cultivars. Another catalog was discovered in 1817, which showed the first description of a striped cultivar. Later cultivars were found in the pages of Carter's Nursery catalog, which shows the first known orange-yellow cultivars, among others. The first blue cultivar was bred by Major Trevor Clark and sold by James Carter in 1860. Between 1860 and 1870, the first known cultivar that produces more than 2 flowers per stem was bred by Stephen Brown. This cultivar was known as Scarlet Invincible. Using this method, others would breed other cultivars with this trait, such as the pink cultivar, known as the Crown Princess of Prussia, Invincible, and Imperial Purple. The first cultivar bred through controlled cross-pollination was the cultivar Invincible Carmine in 1883. This was a crossbreed between a purple and red cultivar. Thomas Loxton and Henry Eckford would take advantage of these methods to breed an extensive number of sweet pea cultivars. In 1901, the Spencer mutation emerged. It was a mutant variant of the cultivar Prima Donna. This trait was found to be mostly stable and was used to breed the Spencer cultivars. The first to discover this was Robert Steinman. This group was named after Earl Spencer, whose gardener discovered this variation. These cultivars produced larger flowers than those that came before. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. The first winter flowering cultivars were made by crossbreeding sweet pea with common vetch. Although they did not take off, these lines were used to crossbreed with those of the Spencer lineage to breed early flowering Spencer cultivars. The Tillamite cultivars were bred from a mutant line of the common sweet pea cultivar, Blanche Fairy. This mutant trait codes for early flowering in non-Spencer lines. The Cupid mutation was discovered in a population of the cultivar Emily Henderson. This mutation causes dwarfing, with the cultivars only being 5 to 8 inches in height. It was discovered in California in 1893 and was quite stable, so it was used to breed other dwarf cultivars. A pink mutation of the same type was also found in a population of Blanche fairy. These two mutations become the foundation of the Cupid series of sweet pea. This series was bred by the Laxton brothers, who bred these dwarf varieties with normal varieties of sweet pea to produce 23 dwarf cultivars. The genetic data, obtained through the various breeding experiments done by Dr. Fleming, found that the Cupid mutation is recessive in nature, and the bright color trait is dominant over the dull flower trait. It was also found that the marble trait is controlled by a single recessive gene. The red flower trait seems to be controlled by two genes. Both are contained within white cultivars. All white cultivars have at least one of the genes, and the interaction between these two complementary genes creates red-colored sweet peas. So if you crossbreed two white cultivars of complementary genes, you'll get a red-flowered sweet pea lineage. It seems to go for all color types, actually. One of the key takeaways from this genetics research done by Dr. Fleming was that all color types of sweet pea are based on the presence or absence of an interaction between two complementary genes. He also found that all rogue plants, previously thought to be mutations of some sort, are actually a product of crossbreeding between genetic lines that happen in the wild, or insufficient seed purification. And that about covers everything. Thank you for watching.